All right, thanks Allison and everyone else. It's super exciting to be here, and I'm excited to tell you about our projects on brain simulation at Google. So what you're seeing on the screen right now is actually a whole brain emulation of a kind, at least, of a small fish, a larval zebrafish, which has been mentioned in Ed's talk, where on the left-hand side is the ground truth, so that's the single-cell resolution neural activity of that fish, and on the right is what a computer program is predicting or producing for a set of activity it's never seen before. So I'm going to tell you the story of sort of how we got to this point. And the general question which my team for the past 12 years has been interested in is, can a computer be programmed to simulate a brain? And we've been trying to build up a set of results, techniques, collaborators, resources, which could systematically make progress on this problem. The first thing we set out to do was try and accelerate Connectomics, which Andrew so beautifully introduced. So this was one of the first things we did was actually work with HHMI Janelia to create the first large-scale map of a fly brain, something called the Hemi brain, which was introduced in 2020. Since then, there have been multiple whole fly brain maps. We also worked with Jeff Lichtman's group at Harvard to produce the first large-scale cubic millimeter petascale map of human cortex. So this is a very different project in which we're doing 1/1,000,000th of an entire human brain, but there's been a lot of interesting science that's come out of this as well. Enabling this progress has been advances in computer science and algorithms, which is, sort of our technical contribution to this field. In 2018, we introduced the first autoregressive technique for tracing neurons, which reduced the amount of proofreading by a few orders of magnitude. More recently, my colleague Michał Czernyszewski has come up with a new approach, which further reduces the amount of proofreading cost by another 2 orders of magnitude, and putting in line with current estimates for acquisition cost. We've also worked with others to use the beautiful expansion microscopy idea and try and bring that into Connectomics. So with Hans Donzel, we introduced a technique called Lycon last year, and we're excited to see many other techniques in this space now developing as well, including PRISM. Okay, but returning to simulation, what do we mean by simulate the brain in this case? So for us, we're going to talk about neural activity. In particular, we want to reproduce neural activity that we observe in some organism. Using all the available data that we can acquire, that could be anatomy, physiology, and so on. And we're going to use AI to fill in missing parameters in a specific way. And there's a commentary if you're interested in sort of more of the philosophy of what we're doing here. So for this, we came up with a project called ZapBench. This was started in 2018 as a collaboration between 2 labs at Harvard, Florian Engert and Jeff Lichtman, and Misha Aren's lab at HHMI Janelia. And the overall question is, given context, given, let's say, the past 30 seconds of neural activity in a fish brain, can we predict the next 30 seconds? And so in order to do this, we had an experimental ambition, which was to acquire both the anatomy and the function from a same specimen, not just the same species, but literally the same fish, and put that into our modeling approach. This is the experimental pipeline of acquiring all that data. Obviously, I'm not going to go into the details, but just to tell you that it took over 100 fish shipping between Virginia and Cambridge in order to get one in which everything worked well enough. So these projects are very heroic on the acquisition side. Finally, though, we got a data set which we were happy with. Here you can see for all 71,000 neurons, 8,000 time steps of neural activity under various different conditions, some of which are more periodic than others. And then we have a training, validation, and test split for all the activity at the bottom. So we take all that activity, and then we train 2 types of computational models. One which just looks at the activity traces. These are sort of classic time series methods for forecasting. And one which actually looks at videos and produces new video frames directly. Here's just an example of what that looks like. So on the left is the ground truth activity. So the blinks are basically neural activity. In the middle is the output of one of our prediction models, and on the right is the difference between the two. So we basically subtract the left and the middle to show you the differences. And I don't know if it's appearing so well here on this screen, but if you want to take a look, find me on my laptop, or I can send you links to the video. And the idea was to put this out as like a benchmark, so anybody can easily access this data set, test their own methods, and see how well they do. One last thing which has been interesting recently is that AI methods are now helping us actually design better methods for simulation itself. So our larger group in collaboration with DeepMind put out a new system which can automatically discover new models for any kind of empirical task, including ZapBench, which it discovered a model that's actually better than any of the baselines that we developed. Others, this was a paper that was just put out a couple of days ago, or at least we discovered a few days ago, in which the researchers invented kind of like a society of agents that collaborate to develop new models, and they were actually able to improve also on the baselines for ZapBench. So I think this is pretty exciting in the sense that we can now very rapidly build complex models from this data and explore the space of models much more quickly. And finally, the most exciting thing in this project, I would say, is all the results I just showed you were just using the neural activity itself. In 2026, we're going to finish proofreading the actual connectome for that specific animal, and then we'll be able to combine those two data sets to have quite an exciting model of a whole brain. Beyond that, we're interested in doing scaling beyond fish, collecting much more multimodal data, and many other things along the way. Finally, I would say just, in terms of how we organize this, we really try to stick to real data rather than anything synthetic, treat everything as a benchmark, and try and generate useful results and also infrastructure along the way. These are the collaborators, which our team has been lucky to work with over the years, so I just want to thank them, and I'll take any questions.
Awesome. Questions in the back.
Thank you for your talk and this great work. I have a question with regards to, you mentioned sort of neural activity and then moving on with the connectome. Are you also eventually including glial selectivity, chemotaxis, just like all the other kind of odds and ends of what could be potentially interesting?
Yeah, I mean, I agree that would be super interesting. Right now, though, we're just focusing on neural activity and trying to get a clean signal of that data set. But in the future, as I mentioned, we'd love to have much more multimodal data sets on both the function and the anatomy side of things.
Hi, I'm Alex. Yeah, not my field, but I'm curious, I guess, if you could dive in a little bit of the philosophical angle, why you want to model. Obviously, there's been a lot of work modeling the connectome and modeling, just neural activity. But is that sort of like where the interesting bits are, or is it, potentially glorified curve fitting? Are we getting qualitative insights about, the nature of the brain's computation out of this kind of work?
Yeah, I mean, I think this is, you kind of have to start with some assumptions. So, for example, you could probably train an LLM just on my behavior, just looking at my behavior for a year or two, and you might get something that actually looks like a simulated me to some extent. But I kind of think that's not that interesting from a scientific point of view. I actually think, neural activity is like sort of the level of abstraction which I would argue the nervous system really cares about. It goes through a lot of effort both below it and above it to sort of to manage activity. So I think if you're going to talk about brain simulation, I really think that's a good spot to try and target.
Yeah, just to follow up though, are you seeing, I guess, say you give the fish various stimuli, obviously you're able to pattern match that quite well. If you give it stimuli that are not directly trained on, are you able to still, does it generalize out of sort of, distribution?
Yeah, that's exactly the kind of thing which we're sort of looking at now, but I wouldn't say we have like a clean result to report.
Very cool, thank you.
Thank you so much, Viren. Awesome. With that being said, Alan, please come to the stage.